Hello everyone, welcome back to another Scientific Weekly. Today we are going to talk about an announcement instead of a discovery or an article, because there hasn't been much interesting this week. So tensions are flaring between scientists that are going public with a plan to build synthetic human DNA. So this started a long time ago, roughly 1990 roughly, so that's about 27 years ago. So nearly 200 leading researchers in genetics and bioengineering are expected to attend to a meeting in New York City coming week and are going to discuss the next stages of the so-called the GP Wide project, or better said, the Genome Project Wide. It's a 100 million US dollar venture to research, engineer, and test systems of building DNA and genes. So this comes forth out of the HGP, Human Genome Project, which lasted for 13 years and ended in 2003, where they fully researched, mapped the genetic code of humans. So the coming up project is actually the next logical step. HGP allowed the scientists to read the genome, but it's still complex to understand. What they are planning to do now is take it a step further. Instead of just reading it, they are trying to write it. This is going to be very complicated because basically it involves life, so to say, because they are going to start writing DNA. The DNA that makes my hands be like this, my face like this, and makes you the way you are. So the synthesis of that can still be redefined to the core. It already exists, but it's complicated, it's expensive, it's not very successful. And as a species, it can transform us, because if this project goes well with time and they discover interesting discoveries, which they probably will, then there might be changes for us, changes that help humanity evolve, changes that prevent autoimmune diseases like asthma or hay fever, for example. They can work against it. They can change our DNA and get rid of it, so to say, which would be very interesting. So now it's mostly still behind closed doors and they are talking because ethics are playing a role. Like I said before, it can have it can mean a lot to the whole human race, and if you keep researching and discover things to do, people are going to be like, hey, why won't we do that? And ethics get into play. Some people be like, yes, let's do it, it's good for humanity. Others be like, don't do it, you are fucking with nature, you don't know what's going to happen, you don't know what change it's going to produce in the end. So it comes to humans, and in that case everyone wants to speak and everyone has its opinion. They are trying to hear that, but on the other hand, science is evolving, science goes further, it just keeps going. You can't say to science, hey, stop. No, science just keeps rolling on its own, no matter what you say. So during the conversation, science is still developing. So they are already working on it, and at the end of this year, 2017, they are hoping to successfully make a synthetic version of a yeast genome. A yeast is mostly used for bread or beer, and it's a small organism. If they can produce a single genome of that, they already have a step. So what are genomes actually? Genomes are, simply said, our genes. Our genes are made up of DNA, and DNA is made up of base pairs. Those base pairs are made up of a single base pair are two separate amino acids, and those amino acids can be combined to make new complications. We are actually a very complicated mixture of amino acids, so to speak. So how deeper you go, how more complex it gets, but also how more complicated it gets. Your understanding grows, but so does the problem. And you can keep going for that actually. So, but the thing is, it's actually possible already to make synthetic DNA, but it's very expensive. Those base pairs I mentioned before for amino acids are 10 US dollar cents a piece. That sounds cheap. Well, let's calculate it to a single human has around 3 billion base pairs. So you're looking at a 300 million price tag roughly to create enough base pair to create enough amino acids of a no $300 million to create enough base pairs to produce a whole human. And then you don't even have all the right combinations in the proper order and stuff like that. Their plans are to reduce this within 10 years by 1,000-fold. That's a lot. So they are currently still running on funding, trying to reach that $100 million target to get it all done. But it's very interesting. There are lots of things they have to think about. The conversation is taking place right now, but at the same time they are also running experiments to make a genome of yeast and then scale it up. Try a plant, maybe a simple organism afterwards, and try human DNA. They are going to scale it up slowly. It's going to take time, that's certain, but how are they going to do it in the end with the HGP project, which is part of the big GP-wide plan? The HGP, it's complicated to say. The Human Genome Project is only one part. They already, like I said before, they are running it with yeast already. Mold might be another step. Plants, simple organisms, and scale it up. They have the confidence and try it with human DNA, so to say. It's going to take a long time. The outcome might be interesting. Ethics are really a big fundamental pillar in this, because you have to cross certain lines. Like, you can't play with human life. In this case they have to for science. So, but on the other hand, you can't change human life as it is. Yes, but the outcome of this research actually might do that already. So, is it good research? Is it bad research? I don't know. I'm torn apart between the ethics and the sheer possibility that might come out of it. So let me know down below in the description or down below in the comments what you think of this. Upvote this video if you liked it, downvote it if you didn't like it. Like I said, leave a comment, subscribe if you didn't do that yet, and if you want to read the whole article yourself, the link will be down below in the description. Thank you guys for watching, and hope to see you guys next time again.